Hello everyone, welcome to Magnet Brains. I'm Aarti and today we are going to study about chapter number 2 from your grade 12 chemistry textbook, that is solutions, and in that we are going to talk about a very important and very interesting topic, that is osmosis. So let's start. First we will talk about the definition and we will see some examples to understand the concept better. So this is the overview of today's class. So we'll talk about osmosis. We'll see what are some examples ताकि हमें काफ़ी अच्छे से समझ में आए पूरा concept. फिर हम osmotic pressure के बारे में भी थोड़ा सा study करेंगे जो कि एक colligative property है. And last में हम लोग देखेंगे what do we mean by isotonic solutions, and we will end the session with a numerical. So let's start. So first, if we read the definition that is given in the book, and I have also modified it a little. So osmosis is a flow of solvent molecules. अब solvent molecules क्या होते हैं? वो molecules जो कि एक higher content में present होते हैं, right? एक higher concentration में present होते हैं. From low solute concentration, solute is that particle which is present in less concentration, कम quantity में present होते हैं. Solvent and solute दोनों मिलके एक solution बनता है. So it is a flow of solvent molecules from low solute concentration to high solute concentration through semi-permeable membrane SPM. So in order to understand this definition, I would like all of you to take this example and imagine. अब जब हम ग्रेप्स लेते हैं एंड उनको हम एयर ड्राई कर रहे हैं, सिंपली हमने उनको एक ओपन कंटेनर में सन के अंडर रख दिया फॉर सेवरल आवर्स. व्हाट डू वी ऑब्जर्व? वी ऑब्जर्व दैट हमारे वो जो ग्रेप्स है वो श्रिंक हो गए हैं, उसमें से वाटर लॉस हो गया है एंड दे हैव श्रंक नाउ, राइट? नाउ विल टेक दोज रेजन्स. अब वो रेजन्स बन गए हैं क्योंकि उसमें से वाटर हमारा इवापोरेट हो गया है. सो अब उन रेजन्स को हम लेते हैं, अगेन फॉर ओवरनाइट वी कीप देम इन वाटर. हम वाटर में उनको रखते हैं फॉर ओवरनाइट. व्हाट डू वी ऑब्जर्व? वी विल सी दैट वो स्वेल अप हो गए हैं, like water has entered into those raisins and again वो हमारे grapes में convert हो गए हैं. But here, do you see any holes in the grapes or any holes in the raisins जिसके through से ये हमारा water या तो बाहर जा रहा है या तो अंदर आ रहा है, right? So this actually is a process which is known as osmosis. But here again two different kind of osmosis are taking place जिसके बारे में हम आज की क्लास में study करेंगे. But actually if you take a grape or you can take raisin also, तो उसमें एक बहुत थिन लेयर होता है, एक बहुत थिन मेम्ब्रेन होता है जिसमें बहुत टाइनी पोर्स होते हैं जिसके थ्रू हमारा वाटर या तो अंदर आता है या तो बाहर जाता है. एंड दैट मेम्ब्रेन, नाउ यू मस्ट बी थिंकिंग, एंड दिस इज वेरी इंटरेस्टिंग पॉइंट ऑल्सो, दैट ये सिर्फ हमारा वाटर को ही अलाउ कर रहा है या तो बाहर जाने के लिए या तो अंदर आने के लिए. अब हमारे ग्रेप्स में या रेजन्स में और भी दूसरे चीज़ें प्रेजेंट होते हैं. फॉर एग्जाम्पल शुगर कंटेंट इज देयर, प्रोटीन इज देयर, डिफरेंट कार्बोहाइड्रेट्स आर ऑल्सो प्रेजेंट. they are not getting lost or getting added. Only water molecule is traveling, right? So इससे हमें ये समझ में आता है कि ये जो semi permeable membrane है, that thin membrane which contains small pores, that only allows water to pass through. जो दूसरे कंटेंट्स अवेलेबल है उनको वो अलाउ नहीं करता, एंड दैट्स व्हाई इट इज नोन एज सेमी परमिएबल मेम्ब्रेन. एंड इनके जो सेमी परमिएबल मेम्ब्रेन के कुछ एग्जांपल्स अगर हम और लें, हमारा जो मैंगो होता है उसमें भी ये लेयर प्रेजेंट होता है. एग हमने देखा होगा, तो एग का जो आउटर शेल है उसके अंदर एक थिन मेम्ब्रेन होता है विच इज अगेन अ सेमी परमिएबल मेम्ब्रेन. सो इट कैन ईदर बी अ नेचुरल, we can get this SPM from a natural source and also from a synthetic source, right? So we will be discussing about those examples as well. So here we will look at this definition once again, और इन examples के through हम लोग इस concept को और अच्छे से समझने की कोशिश करेंगे. So I have mentioned here two different definitions. दोनों ही correct हैं. जो भी आपको easy लगता है उसको हम अच्छे से समझ लेंगे, notes में लिख लेंगे. So I will read the other definition now. It is the movement of solvent molecules from high concentration to low concentration through SPM. Movement of solvent molecules, which is your water. So water कहाँ से कहाँ move कर रहा है? Water is moving from high concentration. So again take that example. Raisin को हमने water में डाल के रखा है. So raisin is having very less concentration of water, right? Because हमने उनको dry कर दिया है. And outside it, जो हमने water लिया है, obviously that is, has lot more solvent molecules. So from the water, that is from the environment, हमारा water कहाँ enter कर रहा है, किस direction में? Inside the raisin, right? So from the higher concentration of solvent, that is water, to the lower solvent concentration, that is raisin. ठीक है? So this is the definition in terms of solvent molecules. Same definition अगर हम चाहें तो हम solute के molecules के terms में भी लिख सकते हैं. See, we know this relation कि अगर solvent ज़्यादा है तो solute कम होगा, अगर solute ज़्यादा है तो हमारा solvent को हम कम मानेंगे, right? और in other terms we can also say कि if the solute concentration is high, जो कि raisins में है, तो raisin के अंदर कैसा atmosphere है? एक high concentration का atmosphere है, right? और अगर हम water का example लें तो water क्या है? उसको हम बोलेंगे कि low solute और low concentration है उसमें, right? So we can just remember it as low solute concentration to high solute concentration. So I have given definition in terms of solute as well as in terms of solvent. So this way हमारा जो osmosis का definition है वो complete होता है and I hope सबको clear हो गया होगा. Now we will be talking about some more examples ताकि हमको ये concept बहुत अच्छे से clear हो जाए. And in exam also, if examples are being asked, then we have ample of examples to write in our papers as well. So let us talk about one experiment which was done ताकि हमें थोड़ा और समझ में आए osmosis के बारे में. So in this, as in the diagram, you can see हमने एक beaker लिया है. In that beaker, it is mentioned here that solvent is present. So solvent, you can just imagine कि हमारा water present है beaker के अंदर. I'll just write here water. And we have taken a thistle funnel. Okay, I have written here also. So thistle funnel क्या होता है? एक हमारा glass, it is made up of glass and it has a long neck. You can see it has a long neck. And at the mouth of this thistle funnel हमने क्या लगाया है? हमने semi permeable membrane लगाया हुआ है. That is your SPM. Now you know कि SPM क्या होता है? It only allows solvent molecules to pass, not any other. So here inside this thistle funnel we have just taken water and mixed some salt or sugar in it ताकि वो एक solution बन जाए. So now just try to think कि हमारा जो concentration है वो किस में ज़्यादा होगा. Very easy to answer, that is inside the thistle funnel because we have made a solution. So हमारा जो concentration है, I'll just try, right? High concentration. And inside this beaker we have only water, तो obviously यहाँ पे जो concentration होगा that will be a lower concentration. So just now we studied कि osmosis का definition क्या होता है? That the movement of solvent molecules from higher solvent concentration to low solvent concentration. So basically यहाँ पे लो कंसंट्रेशन मैंने लिखा तो इसका मतलब है दैट हियर no solvent is present, right? Actually in the experiment no solvent solute is present. So from the beaker the water molecule will move inside the thistle funnel, and here हम लोग देख सकते हैं in the experiment that we observed that the water level has risen up inside the thistle funnel. ठीक है? So using this experiment also we can easily understand कि osmosis क्या होता है and the direction or the movement of particle कहाँ से कहाँ होता है. So if we'd like to read it, so इस experiment में what do we observe? Just to summarize, level of solution rises in the thistle funnel due to osmosis of solvent, or you can simply write due to osmosis. That will also be enough. Now that हमने experiment देख लिया, we have also talked about the definition. We will see some more examples which are actually quite important. So let's talk about those examples now. All right. So before moving to the examples, SPM का एक बार definition लिख लेते हैं, and we'll also see that what are the other examples of SPM. And here I've also put a GIF so that you can understand, better. हम लोग imagine थोड़ा better कर पाएंगे कि कैसे हमारा movement हो रहा है. I think I don't need to explain it because वो काफी easy है to understand, but we'll come to that later. Before that, just write down the definition of the SPM, that is semi-permeable membrane. क्या होता है ये? We have studied कि ये सिर्फ हमारा जो solvent molecules हैं उनके movement में help करता है या उनको allow करता है pass होने के लिए. एक बहुत easy example जिससे हम बहुत easily relate कर सकते हैं. Like if we take some sand और उस sand में हमारे छोटे मोटे pebbles भी present है, and we take that and if we just do filtration using a sieve, so what do we notice कि जो हमारे pebble के size है वो तो it is a bigger size as compared to the sand particles. तो हमारा sand particles तो वो sieve से वो छन्नी से easily बाहर आ जाता है, but जो pebble का size है वो allow नहीं करता कि वो pore से pass out होके बाहर आ जाए. So this is exactly what is the function of your SPM. So semi-permeable membrane भी क्या कर रहा है? ये हमारे सिर्फ solvent particles को pass करने के लिए allow करता है, but जो solute particles है जिनका size बड़ा होता है, then the pores present in the SPM, it does not allow. Very easy to understand. Now moving to the definition. So it is a type of plasma membrane जो कि हमारे हर एक सेल में प्रेजेंट होता है. प्लांट सेल, एनिमल सेल, दे हैव दिस थिन लेयर ऑफ प्लाज्मा मेम्ब्रेन which is SPM, which can allow only solvent molecules to pass through. You can see some examples. यहाँ पे हमारे कुछ natural examples हैं और कुछ man-made या synthetic examples भी हैं. So like I said, egg membrane is there. Cellulose acetate is there. This is known as chemical SPM, so you can write this also. Then we have parchment paper. तो जो baking में भी हम लोग एक baking paper use करते हैं, that is also known as parchment paper. Then we have cellophane membrane, right? So we have much, more examples are there. But I think these are enough to understand कि क्या हमारे natural sources हो सकते हैं. Also remember the example of raisin, mango का भी example हम देखेंगे. So these are some examples of your SPM, natural as well as synthetic. Now moving to this GIF, very easy to understand. यहाँ पे जो ये small blue color का circle है, that represents water molecule, which is your solvent molecule. And this bigger green circle ये हमारा sugar जो कि हमारा solvent molecule को represent करता है. And here in this beaker एक partition है, and that partition is nothing but हमारा SPM है. SPM को हम semi permeable membrane बोल सकते हैं. We can also call it selectively permeable membrane. So here यू कैन सी कि अगर हम दोनों सॉल्यूशंस को साइड बाय साइड रखते हैं, देन ऑटोमेटिकली what word can we use for such a process? हम लोग बोल सकते हैं that it is a spontaneous process, यानी कि ये अपने आप होता है, हमें कुछ करने की जरूरत नहीं है. We keep these two solutions next to each other and बीच में एक SPM हो, then osmosis will occur naturally. That's why we use this word that it is a spontaneous process. हमको कुछ करने का जरूरत नहीं है. And here we will see that low sugar concentration, और ये सिर्फ water है, यहाँ पे कोई भी solute present ही नहीं है. So from low concentration, this is nothing but your low concentration, and the water molecules are traveling to a higher concentration, right? Higher concentration क्यों? क्योंकि वहां पे sugar molecules are also present. That is your solute, right? So I think this is clear to all. Now let's move to some examples. So first we'll try to understand all of these examples through these images, and after that हम लोग one by one will read also, just to understand कि हमारे textbook में भी क्या mentioned है. So first one I would like to talk about is the raisin one. तो क्योंकि हमने ऑलरेडी डिस्कस कर चुका है, सो इसको हम रिपीट नहीं करेंगे, बट दिस इज वन ऑफ द एग्जांपल्स जहाँ पे हम ऑस्मोसिस को ऑब्जर्व कर सकते हैं. वन मोर थिंग आई वुड लाइक टू ऐड, इनिशियली ऑल्सो आई सेड कि जब ग्रेप्स को हम ड्राई कर रहे हैं, सो व्हाट डू वी ऑब्जर्व? वी सी कि वाटर हमारा बाहर आ रहा है फ्रॉम द ग्रेप्स, बिकॉज हम उसको सन ड्राई कर रहे हैं, सो ऑटोमेटिकली व्हाट विल हैपन that the water present inside the grapes, it will come out. So it is coming out. So that will be known as exoosmosis. Okay, it is coming out, so exoosmosis. In this image, we can see that raisins are kept in water. So what will happen after some time? We'll observe कि वो swell हो गए हैं. क्यों? Because water has entered these raisins via SPM. So this is known as एंडोऑस्मोसिस, क्योंकि वाटर यहाँ पे अंदर एंटर कर रहा है, राइट? सो दैट इज एन एग्जाम्पल ऑफ एंडोऑस्मोसिस. आई होप दिस इज क्लियर. नाउ मूविंग टू द नेक्स्ट एग्जाम्पल, काफी इंटरेस्टिंग एग्जाम्पल विच इज ऑफ दिस पिकल, मैंगो पिकल. आई एम श्योर मैंगो पिकल बहुत लोगों ने खाया होगा, बनाया होगा, बट हियर what are we trying to understand? We are trying to understand कि ये क्या प्रोसेस यहाँ पे हो रहा है. जो हमारे पिकल्स की शेल्फ लाइफ है वो क्यों ज़्यादा होती है नॉर्मली, देन दोज प्रोडक्ट्स जिनको हम पिकल नहीं करते हैं, राइट? सो हियर इन द मैंगो पिकल का प्रिपरेशन में व्हाट डू? वी ऐड अ लॉट ऑफ सॉल्ट, राइट? हम लोग सॉल्ट को एज अ प्रिजर्वेटिव ऐड करते हैं. एंड एग्जैक्टली व्हाट इज हैपनिंग, जस्ट ट्राई टू इमेजिन कि मैंगोज के बाहर हमारा कंसंट्रेशन क्या है? सॉल्ट का ज्यादा है, राइट? सो इट इज अ हायर कंसंट्रेशन एनवायरमेंट आउटसाइड इट, बट व्हाट इज द कंसंट्रेशन इनसाइड दीज मैंगोज? लो कंसंट्रेशन. ऑब्वियसली मैंगोज के इनसाइड तो हमारा सॉल्ट कंसंट्रेशन या other concentration would be less, right? So outside is high, inside is lower concentration. So यहाँ पे water movement क्या होगा? From low concentration to high concentration, right? Low solute concentration to high solute concentration, right? So जो भी water present है हमारे mangoes में वो बाहर आ जाएगा because हमने बहुत सारा salt add करा था. And in this way, very interesting question here, जो हम सबको पूछना चाहिए अपने आप से कि इनकी शेल्फ लाइफ कैसे इंक्रीज हो जाती है. जस्ट बाय एडिंग सॉल्ट इनका शेल्फ लाइफ कैसे इंक्रीज हो गया? वन थिंग हियर इज बिकॉज हमने वाटर को सारा रिमूव किया है फ्रॉम दीज मैंगोज, सो द वाटर एक्टिविटी हैज बिकम वेरी वेरी लेस. तो इससे क्या होता है कि जो हमारे बैक्टीरियाज हैं उनको इनफ वाटर नहीं मिलता टू ग्रो, टू मल्टीप्लाई. एंड द सेकेंड पॉइंट हियर इज कि हमारे बैक्टीरियाज को भी ये एनवायरनमेंट मिलता है. तो ऐसा ही एनवायरनमेंट हमारे बैक्टीरिया को भी मिलता है, राइट? तो वो बैक्टीरिया के इनसाइड भी क्या है, लो सोल्यूट कंसंट्रेशन, एंड आउटसाइड क्योंकि इतना ज्यादा सॉल्ट हमने ऐड किया है, सो दैट इज हाई कंसंट्रेशन. सो बैक्टीरियाज भी क्या एक्सपीरियंस करते हैं, ऑस्मोसिस. सो दे ऑल्सो बिकम shrunken, right? So that is the reason कि वो बैक्टीरियाज भी किल हो जाते हैं, हमारा वाटर एक्टिविटी भी रिड्यूस हो जाता है, एंड हमारे पिकल्स की जो शेल्फ लाइफ है वो इंक्रीज हो जाती है. सो इन दिस वे हमारे पिकल्स में यू विल ऑब्जर्व कि दे हैव shrunk and also उनका जो शेल्फ लाइफ है वो इंक्रीज हो गया है. सो दिस इज अगेन वेरी इंपॉर्टेंट एग्जाम्पल ऑफ ऑस्मोसिस. Coming to the next, we can say that, you know, kind of a utility of osmosis in, medical field. यहाँ पे these are the red blood cells which I'll just write RBCs. So हमारे जो red blood cells हैं, अगर उनको भी हम ऐसे environment में रखें, we have taken a beaker and in that we have added some amount of salt. तो वो हमारा solution हो गया. And we put these red blood cells. So basically जो हमारा container है उसमें जो salt का concentration है that should be greater than 0.9% mass by volume, right? 0.9 grams of salt present in some amount of your water. Right, so that becomes 0.9% of m by v, mass by volume. So what will we observe? Again, हमारे जो red blood cells हैं, they have low concentration, outside is high salt concentration. So again, water हमारा low concentration से high concentration पे move करेगा. So we will observe that the red blood cells have become shrunken. Right? So this is the third example. Now, talking about the last example, and this is also very important. There is this medical condition जिसे doctors बोलते हैं oedema. Okay, it is pronounced as oedema. And in this, what happens that water retention can be observed in the tissues. अब इसका मतलब क्या होता है? So, those people who take lot of salt, जो बहुत ज़्यादा नमक use करते हैं अपने खाने में, so what happens? Now that food is being digested, assimilated, और वो खाना हमारे हर एक टिश्यू, सारे सेल्स को पहुँचता है, right? अब जैसे ही that is reached to all the tissues and that is having a high solute concentration. So what will happen here again कि हमारे cells के अंदर ये high concentration पहुंच जाएगा, and outside it what is there? Outside तो we have a normal environment where we can say कि outside there is low concentration. So inside it has become high. So again from low to high there will be movement of water. So what will happen? हमारे cells swell up हो जाएंगे. So that person will again, look like कि swelling आ गया है, right? So why is that happening? Because जो भी हमारे body के tissues हैं, cells हैं, वो सब swell up हो गए हैं because of osmosis, and this is a medical condition which is known as oedema, right? So this becomes, our last example to understand what is osmosis. Now, moving forward, again यही सारे examples जो अभी हमने images के through पढ़ा, इन्हीं में, and I think few more examples I have added here. We'll just read. So the first one is about the raw mango. So here a raw mango placed in concentrated salt solution, क्योंकि हम pickling कर रहे हैं, right? Loses water via osmosis, वो water को lose करता है, and they become shrunken and shrivel into pickle. This is how pickle formation takes place. Second examples, I think ये भी हम सब ने observe किया होगा. That is wilted flowers, they revive when placed in fresh water. Now अगर हम रेगुलर वाटरिंग नहीं करें हमारे प्लांट्स में, सो वी इजीली ऑब्जर्व कि वो काफ़ी मुरझाए हुए हो गए हैं, उनके लीव्स भी हमारे काफ़ी मुरझाए हुए हो गए, राइट. बट जैसे ही वी प्रोवाइड वाटर, रेगुलर वाटरिंग, देन दे विल ऑलवेज लुक वेरी पर्की. जैसे because again what is happening here, osmosis is taking place. So when we are providing water to the roots, तो हमारे roots के through water travels up to each and every leaf, to each and every flower, जिसके वजह से वो काफी fresh and revived दिखते हैं, right. Again if we don't provide water, so वो मुरझा जाएंगे. That is also because of osmosis. एंड हमने ये बहुत बार देखा होगा कि जब जो सब्जी वाले रहते हैं वो क्या करते हैं, वो रेगुलरली जो भी हरी सब्जियां मोस्टली, एंड आई हैव रिटन एग्जांपल ऑफ कैरेट, तो उसमें भी क्या करते हैं वो, दे कीप एडिंग वाटर, स्प्रिंकलिंग वाटर ओवर देम, राइट. एंड व्हाई इज दैट, ताकि वो फ्रेश दिखे, राइट. हम लोग बार बार उन पे वाटर छिड़कते रहेंगे, सो दैट वे दे विल नॉट लुक वेरी ड्राई, वेरी मुरझाए हुए, राइट. सो अ carrot that has become limp, that means it has lost its stiffness, we can say, because हमने water उस पे नहीं sprinkle करा, because of water loss in the atmosphere. So continuously because of evaporation हमारा water loss होता है, can be placed into the water, making it firm once again. So the same example, if we sprinkle water or if we keep these vegetables or fruits in water, then they will be fresh again. Water will move into cells through osmosis, just as हमने देखा कि अगर हम लोग किसी भी प्लांट को वाटर करते हैं, so through roots water is reached to each and every leaf. Then the next example is about the blood cells. So blood cells, when placed in water containing less than 0.9 m by v is what? Mass by volume. ये क्या है? ये एक हमारे concentration का term है. Salt, blood cells swell up, right? So this becomes an example of what? अगर बाहर हमारा low concentration है, inside the cell there is a higher concentration. Usually हमारे जो blood cells है उनमें 0.9% जो salt solution होता है उसके equal concentration होता है. That's why we are taking 0.9 as kind of standard here, right? तो वो swell up हो जाएंगे because lower concentration to higher concentration हमारा water move करेगा. And they collapse, यानी कि वो shrink हो जाते हैं again जब concentration हमारा ज़्यादा होता है, then 0.9% m by v mass by volume. तो बाहर concentration ज़्यादा है, inside cells कम है, so again what will happen? They will become shrink. Now the last example which was of oedema, that is again people जो बहुत ज़्यादा salt अपने खाने में लेते हैं, so they will experience this medical condition because of water retention, right? That is known as oedema. So I hope all of these examples are clear to you, and all of these examples are there in our textbook as well. So these are quite important examples. Now let's move to the next topic. So this is again very interesting topic, but इसको समझने से पहले हमें पता होना चाहिए what is osmosis. So that's why we covered osmosis first. So here let's talk about osmotic pressure, which is denoted by letter pi. Okay, so this represents your osmotic pressure. Now let's take an example to understand this osmotic pressure. Now let's imagine that I and you, we are there in a room, and if I keep the door open of that room, then it will be very easy for you to come in, right? Because door is open and you can easily walk inside the room. But if I close that door and if I apply some kind of pressure on that door, ताकि मैं ब्लॉक कर सकूं आपका एंट्री को, सो व्हाट विल हैपन? इसमें दो केसेस हो सकते हैं, या तो यू अप्लाई वेरी हाई प्रेशर एंड यू जस्ट कम इनसाइड, और इफ द प्रेशर जो मैं अप्लाई कर रही हूँ एंड द प्रेशर यू आर अप्लाइंग इज इक्वल, सो दैट आई हैव स्टॉप्ड यू फ्रॉम कमिंग इनसाइड, right? So this exactly is happening in osmotic pressure. So we can imagine that I am the solution. Solution में solute and solvent दोनों present होता है, and the other side of the door we have solvent, right? So where no solute is present basically. So movement क्या होगा osmosis का हमारा यहाँ पर है एक high concentration और वहाँ पे है low concentration. So low concentration to high concentration will be the movement, right? उस तरफ से there will be movement of solvent molecule. So if we provide that amount of pressure in order to stop you from entering, कहाँ पे apply करेंगे? To the solution side, then I can stop the flow of water molecules. I can stop you coming inside, right? So this is the osmotic pressure. तो यानी कि ये वो एक excess of pressure होता है जो कि हम लोग solution side में apply करते हैं ताकि हम लोग osmosis को रोक सके या जो movement है solvent molecules का उसको रोक सके, right? So that is known as osmotic pressure, right? So this way, let's read the definition here, then we will understand this using an example also. So it is excess of hydrostatic pressure which is developed over the solution column due to osmosis. So this is one of the definitions. Now coming to the next definition here, which is it is the external pressure which must be applied over the solution column through the weightless piston in order to stop the flow of solvent inside the solution. So हमें कितना pressure apply करना पड़ेगा solution side में जितना concentration है, right? अगर concentration कम है, so we'll have to apply less pressure in order to stop. अगर concentration ज़्यादा है, so we'll have to apply a higher pressure in order to stop. So this way हम क्या समझ पा रहे हैं यहाँ पे? That the osmotic pressure is directly related to the concentration, right? That is the number of solute particles, and hence this is a colligative property जो कि हमारे number of solute particles पे depend करता है and not on the nature of the solute. Let's see an experiment to understand osmotic pressure better. So here we have taken two containers and दोनों ही हमारे SPM के through connected हैं, right? So here we can see that one side we have taken solution जिसमें solute is there and solvent is there. On the other side we have taken solvent. So वहाँ पे only water molecule is present, no solute particle is present. So जितना concentration इस solution का होगा, that means अगर solute का अमाउंट ज़्यादा है so high concentration, solute का अमाउंट low है तो low concentration, right? So what will be the movement here actually from low concentration to high? So from here there will be movement of water molecules. So हमें क्या करना है? हमें stop करना है इस movement को. So what are we going to do? We are going to apply some amount of excess or external pressure in order to stop this. So हमने ये pi, that is osmotic pressure, ये apply करेंगे on this is your piston, movable piston. So इसके through हम इसके ऊपर pressure apply करेंगे. कितना apply करेंगे? जितना इसका concentration रहेगा उतना. Like if the concentration is low, then low pressure we can apply. If high, then high pressure. We'll apply this high pressure and eventually we'll be able to stop the flow of this solvent molecule from the solvent side, right? Easy to understand. हमें क्या करना है? हमें excess of pressure apply करना है on the solution side ताकि हम लोग जो मूवमेंट है सॉल्वेंट का उसको रोक सके, एंड दैट एक्सेस ऑफ प्रेशर इज नोन एज ऑस्मोटिक pressure, which is a colligative property. So if we'll just go through the definition here, already हमने पहले cover कर लिया, but once again, the excess of pressure जो कि किसके equal है, जो कि हमारे osmotic pressure के equal है, it must be applied on the solution side to prevent osmosis, right? Osmosis को stop करने के लिए we are applying this pressure, which is known as osmotic pressure. Now moving forward, just to summarize कि हमारा what is happening in the osmosis basically. So we take pure solvent and solution on the other side, and they are separated via a semi-permeable membrane, but after some time हम क्या notice करते हैं, from low concentration to high concentration movement takes place, and this is known as osmosis. In order to stop this movement, what will we do? We will apply some external pressure on the solution side ताकि हमारा osmosis stop हो जाए, right? So this is your osmosis and your osmotic example. So just a summary here. Moving to the last topic, that is isotonic solutions. So iso word का मतलब होता है same, right? And tonic, it represents for your concentration. So those solutions जिनका concentration same होता है, जिनका concentration equal होता है. Now imagine अगर ऐसे solutions को हमने SPM के through contact में रखा है, then what will happen? Will there be any osmosis? No, right? Because दोनों ही solutions में हमारा जो concentration है that is same, so there will be no flow of solvent molecule, right? And also one important point that you should remember here, that is कि ये जो osmosis है ये कब तक होता है? This osmosis will happen only जब तक दोनों तरफ हमारा concentration equal नहीं हो जाता, right? Till then there will be movement of solvent molecules. हम लोग बोल सकते हैं जब तक equilibrium का state नहीं आ जाता, after that osmosis will stop on its own because it is a spontaneous process, right? If we'll just read the definition, it's very easy to understand. If two solutions जिनका osmotic pressure same है at a given temperature, then these solutions are known as isotonic. When such solutions are kept in contact through SPM, then no osmosis is observed. और ऐसे solutions को हम isotonic solutions बोलते हैं. See, in this example, in this image also, you can see कि दोनों ही तरफ जो हमारा concentration है वो equal है. So there is no net flow basically. हम बोल नहीं सकते कि there will be any movement. There will be no movement. Such solutions are in equilibrium, so no osmosis will take place. Example. So for example, 0.90%, which is your mass by volume, ठीक है, 0.90 grams per 1 liter of solution, we can say, of your NaCl solution is called physiological saline solution, because similar concentration हमारे cells tissues के अंदर भी होता है, 0.9% mass by volume, and it is isotonic to blood cells. So अगर हम blood cells को 0.9% m by v salt solution में रखते हैं, then there will be no movement of solvent molecule. That is known as isotonic solution. Let's move forward. In this image, we can understand the three types of solutions जो कि हम osmosis के अंडर ही स्टडी कर रहे हैं. फर्स्ट एग्जाम्पल हियर is of hypotonic solution. So in this, what do we see? कि outside जो है वो क्या है? The concentration is less, right? So outside the concentration is less than 0.9%. Weight by volume लिख सकते हैं हम लोग यहाँ पे, mass by volume भी लिख सकते हैं, right? So what will happen here? Endo-osmosis. The water will come inside the cell, क्योंकि outside concentration हमारा low है than 0.9, so what will happen? Low concentration to higher. Endo-osmosis, enter कर रहा है water, right? And this is also known as hemolysis, right? तो ये हमारा biology से related term है, but we should remember that also. Second condition is of isotonic solution, where the concentration inside the beaker is also 0.9 mass by volume. So no osmosis will occur. This is isotonic. Now third condition is of hypertonic. So hyper में क्या हो रहा है? Hyper means, you know, high, more concentration. So outside the concentration is greater than 0.9%. So what will happen? Low concentration से higher concentration. So inside the cell low. So अंदर से बाहर हमारा water का movement होगा, and this is known as exoosmosis. One more term that we should remember, that is plasmolysis. So all of these are important for us to understand. ये सारी चीज को हम you can write it down in your notes so that we remember it. Now let's move forward. Very important point here. So अगर हमारे numericals में, questions में, they are discussing about isotonic solutions, so we can use this formula directly and we will get right answers. So what is happening in isotonic solutions? हम बोलते हैं कि osmotic pressure क्या है? Equal है दोनों solutions में. That's why osmosis is not happening. So we can write pi 1 is equals to pi 2. इसका मतलब क्या है कि दोनों ही solutions में जो concentration है वो same है. So we can write C1 equals to C2. अब concentration का मतलब क्या है? Molarity. Okay? Molarity is our concentration term, and molarity का मतलब क्या होता है? Number of moles of solute present in a liter of solutions. So वही चीज हमने यहाँ लिखी है, N1 by V. N1 represents number of moles. V1 volume equals to N2 by V2. And now we are just assuming कि हमारे जो दोनों volume है दोनों solutions के, for example हमने we have taken 1 liter only, right? Volume. So both your V1 and V2 becomes equal. So we can just write V here. And what is the equation now? It is N1 equals to N2. Now what is the formula of number of moles? So number of moles is nothing but weight in grams of solute. B represents solute divided by volume in liters, right? तो वही चीज यहाँ लिखिए, W1 by M1 equals to W2 by M2. So this is the formula which we can directly use अगर question में isotonic solutions को discuss कर रहे हैं. Let's see a numerical to see how are we using this formula. Right. So let's do this numerical. So the question says 4% of sucrose. Sucrose का formula क्या होता है? Molecular formula. This is our common table sugar. So the formula of this is your C12, H22 and O11. So इसका अगर हमें molecular mass निकालना है, then हम कैसे निकालेंगे? See, we have 12 carbon and for 1 carbon the molecular mass is 12 grams per mole, right? So it will be 12 into 12 plus, for hydrogen it is 1. So 1 into 22. Oxygen, for 1 oxygen it is 16. So for 11 it will become 16 into 11. If you solve all of this, you will get the answer as 342 grams per mole. Right? So this is the molar mass of your 1 mole of sucrose. इसको अभी के लिए मैं erase कर रही हूँ. Then we'll write the formula and complete this last question. So we can write that pi 1 is equals to pi 2. That is isotonic के लिए हम ये लिखते हैं that the osmotic pressure is same. So we can understand that दोनों का concentration भी क्या है? Same है. And if we just come to the formula here, that is your number of moles N1 divided by your volume. I am assuming कि दोनों का volume जो है वो क्या है? Same है. So here your volume will get cancelled and we will only be left with N1 is equals to N2. Now our formula will look like something like this, that weight in grams of first, that is हम sucrose मान लेते हैं 1 को, divided by its molar mass is equals to weight in grams of that unknown organic compound divided by its molar mass. So W1 का value क्या है? Because here हमको दोनों ही values percentage में given है, then no need to divide it by 100. We can simply write 4 divided by the molar mass of sucrose 342 equals to W2, that is 3%, and we have to calculate the value of M2, right? So if we just cancel this, then here it will go by 8, then 5 and 0.5, right? So your M2 value finally we'll just have to multiply 3 into 85.5. This will come out to be here we will have 25. This is molar mass, so we can just write the unit also, that is grams per mole. So 256.5 grams per mole is the molar mass of the organic, that was unknown organic compound. Alright, so with this we have finished our numerical. I will erase this and I will show you the solution here. So you can note down this solution if that was not clear to some of you. And we are done for today's class. So basically we discussed about osmosis, examples of osmosis, then what do we mean by osmotic pressure, then उसके लिए हमने एक experiment को भी study करा, then we discussed about isotonic solutions. We also saw different types of solutions that come under osmosis and the formula that we can use for isotonic solution, and lastly this one, numerical. So thank you everyone for watching and have a nice day. Bye-bye.